Yeah, good to go.
Yes, I think we should begin with company
So,
presentation and part of
so
presentation.
uh, I welcome all of you, all the eminent speaker we have here today. Today evening, you've taken time off to attend. I mean, to conduct this webinar and on an important topic like Urticaria Decoded. So, I welcome all the audience who have already joined us much before the time. I mean, there are a huge number of people from across the country, from Kashmir to Kanyakumari, from Gujarat to Northeast, who have joined us, all eminent dermatologists in the audience. And today's topic is very interesting. And it is my privilege, before I share introduce the speakers and the faculty, it is my privilege to share with you one corporate, a short presentation on Menarini.
Asia, where opportunities abound and the fastest economic growth achieved, is also witnessing the fastest growth in the healthcare sector, which is why we've established our presence and positioned ourselves as the partner of choice to bring important healthcare brands across the region. Menarini Asia Pacific. As part of a leading global biopharmaceutical company, Menarini Asia Pacific's capabilities span the entire pharmaceutical development life cycle. A fully integrated biopharmaceutical company capable of research, development, manufacturing, regulatory affairs, and commercialization. Beyond end-to-end capabilities and cross-functional expertise, Menarini Asia Pacific has also become the preferred partner for leading healthcare companies around the world. Our expertise in building healthcare brands, flexible business model, and dedicated alliance management to create value for our partners have enabled us to make strategic acquisitions and licensing arrangements, consequently taking many brands to greater heights. Our rapidly growing businesses are bolstered by over 3,300 sales, marketing and support professionals operating in 13 key markets. Our sales and marketing strategies, underpinned by local patient and physician insights, have helped many companies with varying needs tap into the Asia Pacific growth story. Whether for new brands or line extensions, pharmaceutical or biotechnology companies can gain rapid market access to the region and manage their risks by leveraging on our strengths as a gateway to Asia Pacific, a reliable partner whose network and reach extends beyond the region, with a strong track record offering unparalleled customer experience. Founded in 1886, the name Menarini has through the generations become synonymous with the mark of quality. A trusted biopharmaceutical company with a strong tradition in partnering, an established presence in over 100 countries with more than 16,000 employees, Menarini is bringing its illustrious heritage and compelling values into the next century. And here in Asia Pacific, our ongoing commitment is to deliver quality healthcare brands, invigorating lives across the region. Your partner, your brand, at your service. Menarini Asia Pacific.
So once again, So once again, I welcome you to all this today's webinar is a series of webinar. Actually, we will have discussion on what, why, which, and about and each. So this is a series in a way that we have planned it on 3 different days with 3 different international speakers. We have today Dr. Marcus Maurer, who is a professor of dermatology, allergology. Will in detail give an introduction about him. Dr. H.C. Thorsten Zuberer, again Professor, Dr. Medical, and Dr. Professor M.K. Church. So we'll have these meetings on 27th of June is today, 8th of August, and 22nd of August. For all the 3 events, we will have Professor Dr. Kiran Godse, who is the Professor of Dermatology, as a moderator. So when we talk about, this agenda of today's, I think it's very well known because we have shared with you, we all have gone through the agenda. So we have Q&A session at 6:20 to 6:35. So request audience to post your questions on the chat, which will be taken by the faculty and the international speaker, during this period. And after the program is over, whatever questions will still be there, they will be answered. So coming to the brief introduction on the speakers, we have today an international speaker, Professor Dr. Marcus Maurer, who is basically from Germany, and he is he has been an associate professor. In fact, he is having multiple roles at the same time. So just associate professor, Department of Dermatology and Allergy, University of Berlin. Then he is section head, vasculitis and connective tissue disease section. Then he's adjunct professor of dermatology and allergology in Beijing University. He's adjunct professor, School of Medical Sciences, Griffith University, Gold Coast, Queensland. He's a membership in scientific societies, Society of Investigative Dermatology, European Society of Dermatological Research, and there's a lot of societies. So other activities, he has done a lot of work in Urticaria network, and in advances in dermatology and allergology, he's on the editorial board member. World Allergy Organization Journal is an associate editor for Europe. Pakistan Allergy, Asthma and Immunology Society is an international advisory board member, and Japanese Society of the Cutaneous Immunology and Allergy, he is a board member. His scientific area of interest are characterization of physiological function of mast cell, neuroimmunology, inflammation, innate immunity, and tolerance. And he has more than 600 peer-reviewed publications, more than 40 books, and the book chapter. So we welcome Dr. Marcus Maurer, who is an international speaker for today's event.
Sanjay, thank you very much for your very kind introduction.
Yes,
Kiran,
we are privileged, and Indian doctors are also privileged to see you among us. Then we have Dr. Kiran Godse, who is MD, PhD, FRCP, Glasgow. He's a professor of dermatology, Department of Dermatology, D.V. Patil Hospital and School of Medicine, Navi Mumbai. More than 105 publications, more than 6 books, 12 chapters. Dr. Godse has been a national IIDVL vice president for 2013, and he was a president IIDVL 2020. Anything connected to urticaria, he is the authority in India, I will say, and done a lot of research, and he was a coordinator for SIG urticaria also. So then we have Dr. D.S. Kripashankar. Dr. D.S. Kripashankar is a consultant dermatologist, MD, DVD, FRCP, Maligi Medical Center, Bangalore, and formerly head of Department of Dermatology, Manipal Hospital, Bangalore, and PGT and examiner, scientific committee chairman. He has again published 60 index publications, 1 book on scoring system, and 7 textbook chapters. He has been invited to more than 298 lectures, and his area of special interest is contact dermatitis, photodermatology, dermatological surgery, lasers in dermatology. And he has, to his credit, Dr. Siddhipa Oration IADVL KN Karnataka 2013 award. Then we have Dr. Abir Saraswat, consultant at Indushri Skin Clinic, Lucknow. He's a dermatologist from a FMC background and with MD, DNB, MNAMS. More than 64 publications in Index Journal, 46 out of which are international, 18 are Indian. More than 100 invited lectures in various conferences. Has the interest lies in the psoriasis, pediatric dermatology, dermatosurgery, alopecia, and dermatopathology. We have amongst us Dr. Vijay Zawar, who is also going to make a presentation, a case study presentation. He's MD, DVD, DNB, FRCP, Edinburgh. Current position is professor and head of Department of Dermatology, VPMC, Nasik, Maharashtra, and he's hold a lot of academic positions in IADVL during 2011 and 12. And it's been a coordinator in SIG Urticaria 12-14 and convener of SIG Pigmentary up to 2021. He has been a president of IADVL Maharashtra in 2015 and also vice president of IADVL in 2017. He has been on the editorial board of National IJD, International Journal of Trichology, and IDOG, JCAS International, JDCR. I mean, a lot of journals to his credit where he is on the editorial board. And he has a lot of awards and scholarships. BM Ambadi Oration 2011, SN Cooper Oration 2017, Ganpati Panjay Award 2021, membership to all international and national societies, PubMed Index publication more than 100, textbook chapters around 14, and again interest of area of interest is rosacea. So it is area of interest is pityriasis rosacea and urticaria axo-oconosis. So with this, I welcome all of you, and I'm sure the audience and doctors who have connected all across the country will find value for their time and on a Sunday evening. And I thank you all for making it to this webinar on. And over with this, I will request Dr. Kiran Godse, who is the moderator for the event, to take the meeting forward. Sir. Thank you, sir. Over to you.
Yeah, welcome. Good evening to all. Happy to see Marcus here. We have got Urticaria Guru here, so I will not spend much of time. Just to tell you that me and Vijay, we had our initial training and learning from Dr. Marcus. So we call him a guru, and he has actually truly long hair. He just to grow beard, that will be true guru with us. Thank you, Marcus, for joining. And we have an interesting session from Dr. Marcus, and then there's a panel discussion. Audience, please send your questions as much as you want, so that we'll try to take it up. So we are delighted and honored that Dr. Marcus Maurer is with us, and he'll be speaking on etiopathology and advances in the diagnosis of urticaria. Over to you, Dr. Marcus.
Thank you so much, Kiran, and thank you to Manorini for doing this. Hello from Berlin. I'm joining you live, and as I'm bringing up my screen, let me just say I'm very happy that we're doing this. These times are difficult for all of us, and to connect and share is very important. And it is meeting like these where we can actually learn about what's new, learn about our difficult patients. So I'm very happy that Manorini is doing this, and I'm even happier that you guys have asked me to start us off with this series, start us off at the very beginning, and that is the new understanding of the etiopathology. What do we know about why people get urticaria, and how does this guide our diagnostic approach to these patients? Only when we know about this can we provide good treatment to patients with this devastating disease. I will give you a sneak preview of the most current version of the guideline. Now, the one we're working with right now is the one that we all worked on in 2016, then finished it in 2017. And you will remember this beautiful on-site, in-person consensus conference in Berlin, We voted with green cards and of course red cards until there were only green cards, until we had a majority to support the recommendations of this guideline. And this was a fantastic meeting, and we really wanted to do this again last year, but things came differently, so we had to be flexible, and we were. And we had a very nice consensus conference, hybrid. So people were in Berlin and people were on their screens voting, discussing, and this 2020 update that will be published soon really is novel in many different ways, including the diagnostic approach to chronic urticaria. So I'm going to highlight for you some of these insights that came from this consensus meeting and that will apply or already apply to our clinical practice in urticaria today. Now, what hasn't changed is the definition of chronic urticaria. Now, we speak about chronic urticaria with wheals, angioedema, or both that occur for longer than 6 weeks, and we still distinguish between spontaneous urticaria and chronic inducible urticaria. In fact, this has become more important. Why? We want to know in each and every patient what type of chronic urticaria we are treating. And we do know today that most patients with chronic urticaria, spontaneous or inducible, have angioedema. And I want us all to recognize and always talk about this. Chronic urticaria comes in 3 different ways. It comes as a patient with wheals, it comes as patients with wheals and angioedema, but it also comes as patients with angioedema without wheals. This is important because as of now, these patients with angioedema only, who also have chronic urticaria, most commonly chronic spontaneous urticaria, they are underdiagnosed and undertreated, more so than the other patients with chronic urticaria, the ones with wheals. We know this, and we know that 70% of patients are affected. 7 out of 10 patients have angioedema, now usually together with wheals, but sometimes without wheals. And this is, to them, a big problem. They have a lot of downside events and effects of having angioedema, including more difficult to treat chronic urticaria and the fear of suffocation, which of course in chronic urticaria does not happen, but not all patients know this. With that backdrop, let us talk about what we know today about why people get chronic spontaneous urticaria, with wheals, without wheals, angioedema, no angioedema. And we like to talk about the things we know. And what we do know is that this is a mast cell-driven disease, where mast cells in the skin get activated, and because of this, degranulate and release histamine and other mediators that then cause itch, erythema, flare, and edema. And with this edema comes the recruitment of cells, explaining all of the signs and symptoms of chronic urticaria, chronic spontaneous urticaria. And the question, what does this? What makes mast cells go from, on the left, not degranulated, resting, to the right, degranulating? This question has been answered. And the answer to this question is the one receptor out of many receptors on mast cells that causes degranulation in chronic spontaneous urticaria. The most important one is the high affinity IgE receptor. And the way this happens is not by an allergic mechanism, for which this receptor, of course, is also very relevant. No, this is a mechanism that uses the IgE receptor in a different way, actually in 2 different ways. I'll share that with you. The mast cells in the skin of our patients with chronic urticaria express, of course, the high affinity IgE receptor. And when we talk to allergy patients, well, then we have to expect that on that IgE receptor, IgE is bound, IgE that is directed to pollen and house dust mite and, well, all the allergens that make our patients with allergies have problems. But this is not the IgE that sits on the IgE receptor of mast cells in chronic spontaneous urticaria patients. No, Their IgE is directed to autoantigens. So it's a form of autoimmunity where the autoantibodies are IgE autoantibodies. And we call this either autoallergy or autoimmunity type 1. Now, there is a type 2, and this type 2 is less frequent, but more troublesome for patients. It is autoimmunity type 2, chronic spontaneous urticaria, where IgG antibodies to mast cells activate these mast cells to degranulate. So these two forms we will find in our patients with chronic spontaneous urticaria, and the guideline knows this. And the guideline also knows that these two forms of chronic spontaneous urticaria, well, they differ. So we should know about this, and we should bring questions and tests on board to our diagnostic approach in chronic spontaneous urticaria. Why? Well, one thing is that if you have a patient with autoimmunity type 1, autoallergy, you can expect different treatment outcome. I'll show you one example here, and that is if you select patients with autoallergy as the cause of their chronic spontaneous urticaria, as was done for this very first study with anti-IgE, omalizumab, then you get a very high response rate. 7 out of 10 patients have no more wheals, 0. This is not what you get when you select patients with type 2 autoimmune chronic spontaneous urticaria. Now, how do we diagnose these patients? You can do what our friend Jorge Sanchez did in Colombia to his patients. He skin prick tested with autoallergens. He used thyroid peroxidase, a very common autoallergen, and he, when skin prick testing his chronic spontaneous urticaria patients, had patients who developed a wheal, showed a response to this autoallergen. In fact, he could, with the blood of his patients, transfer this reactivity to his own skin, showing that it is IgE to thyroid peroxidase that drives whealing in these patients. Now, this is proof of concept, but not something that we can do in our clinical practice, right? We're not going to do skin prick testing with thyroid peroxidase and with human interleukin 24 or any of the other autoallergens. We need to be able to identify patients with either type 1 autoimmunity or type 2B autoimmunity by different means, something that's easier than doing the actual immunoassay or doing skin prick testing with autoallergens. And the same really applies for that difficult to treat subpopulation of autoimmune chronic spontaneous urticaria, the type 2, type 2B patients. Now, in big centers for urticaria, UCARES, it is very likely that many tests that are required to diagnose true type 2B autoimmune chronic spontaneous urticaria are available. What do you need? You need the autologous serum skin test. You need a basophil test, could be a basophil activation test or a basophil histamine release assay. You need an immunoassay for these IgGs to the IgE receptor or IgE. So you need 3 tests. In fact, when you use these 3 tests in your patients, you will find that about 1 in 10 patients is triple positive, has true type 2B autoimmune chronic spontaneous urticaria by the book, by the definition. Now, this interesting study, which allowed for the distinction between autoallergic and autoimmune chronic spontaneous urticaria, also showed something else. And it put in context what we see in clinical practice a lot, where patients come to us and they think that they know what drives their chronic spontaneous urticaria, either stress or infections or drugs that they take, medication that they take, or food. No? How often does that happen to you? It happens to me every week. Doctor, I think it's what I eat. Now, what I do to explain this is we know where chronic spontaneous urticaria comes from. It's an autoimmune disease, autoallergic or true type 2B autoimmunity. But we also know that these things that our patients notice as aggravators of their disease, for example, stress or food or drugs or infections, that they translate to signals in the skin of these patients that modulate mast cells to respond Well, more easily to these autoantibodies. So it's really two things that come together. On the one side, we have signals that make mast cells twitchy, the modulators. And on the other side, we have signals that make mast cells degranulate. And the more of both you have, well, the more likely you are to have a patient with a lot of problems. Bringing all of this on board, what diagnostic measures should be considered in the workup of chronic urticaria? And the current guideline, the one we're working with right now, says we recommend limited investigations. No, the days of doing 100 tests are over. No good, delays treatment, costs too much, and usually doesn't bring a solution that is better than what we do to all the patients anyway. So basic tests that are recommended include a differential blood count, C-reactive protein, and erythrocyte sedimentation rate. That's the current guideline. No, further examinations are recommended, but only based on the history of patients and where indicated. So, and to tell you what the new guideline says, this stays. No, this stays. But there is something else. And I'll come to that in just 1 minute. The current guideline also says we do this to exclude differential diagnosis, that's number one, to rule out severe inflammation, and we also bring on board measurements of chronic spontaneous urticaria disease activity, impact, and control. The new guideline goes further, and the new guideline recommends to do 7 things, sometimes referred to as the 7 C's of chronic spontaneous urticaria: confirm, cause, comorbidities, consequence, cofactors, components, and course. Let's walk through them. You'll find this easier than it looks at first sight. It's straightforward, and it brings on board the important questions that we are to ask our patients and the resulting tests that we should perform. We start with differential diagnosis, but we have to recognize that the whole concept, the whole layout is still, we ask a question, we get an answer, and based on the answer that we get, we either decide to do testing or to not do testing. So let's walk through this for the 7 C's of the diagnostic workup in chronic spontaneous urticaria. Now, confirm. We do ask questions because we want to know, is this really chronic spontaneous urticaria, or could this be urticarial vasculitis or an autoinflammatory syndrome, or in patients who only have angioedema, could it be hereditary angioedema? These questions will guide our testing, whether to do it or not, and if so, if we do testing, which one? Cause. Well, we discouraged from looking for the cause of chronic spontaneous urticaria, because what did that mean? In the past, it meant that we did useless allergy testing and wasted time and money and expectations and hope in our patients, where this will not bring a positive outcome. Today, we say we know at least 2 causes of chronic spontaneous urticaria: autoallergy and autoimmunity. So let's look for these causes as best as we can. And we know that when we look at these causes of autoimmunity, type 1 and type 2B, or again, autoallergy or autoimmune, then identifying these patients in these 2 groups comes with a different profile that these patients show us. Let's look at that. On the right, you have these type 1 autoimmune chronic spontaneous urticaria patients, autoallergy. They have a higher rate of allergy comorbidity, and importantly, a high rate of response, quick response to omalizumab. The less frequent patients with type 2B autoimmune chronic spontaneous urticaria, true autoimmunity, they also have their signature. They more often have other autoimmune comorbidities. They more often have other autoantibodies, such as ANAs or thyroid antibodies. They have a longer disease duration, more severe disease, more often angioedema. signs of inflammation more commonly, and this is important, a different response to treatment. And this applies to both first and second line treatment, antihistamines, and third line treatment, omalizumab. So it is important to figure out which one is an autoallergic patient and which one is an autoimmune patient. And okay, you can do all of these three tests if you have them: immunoassay, basophil testing, and autologous serum skin test. Or you can use the biomarker that is recommended, will be recommended by the new version of the guideline. A combination really of two tests. Number one, anti-TPO, IgG anti-TPO. That's the regular thyroid workup that you do if you suspect Hashimoto thyroiditis. Anti-thyroid antibodies. Why? Because patients who have elevated anti-thyroid antibodies are more likely to have autoimmune chronic spontaneous urticaria. And here's the kicker. The second one you're going to measure is IgE. Why? Patients with autoimmune chronic spontaneous urticaria more often have low levels of IgE. And a patient with low IgE is more likely to have autoimmune chronic spontaneous urticaria. Now put these two together. You have a patient with one, high IgG anti-TPO plus two, low IgE, and you have your autoimmune chronic spontaneous urticaria patients. The best biomarker is the combination of both, high IgG anti-TPO plus low IgE. You can all do this. So the guideline recommends that we all do this. And at least in a specialist setting, patients should be assessed for, well, the baseline, CRP, ESR, differential, and this marker that helps us to tell apart autoimmune and autoallergic patients, IgG anti-TPO and IgE. Now, we've looked at the cause. Let's look at the comorbidities. This is important. Why? Because many patients with chronic spontaneous urticaria also have chronic inducible urticaria, also have another autoimmune disease, and 1 in 3 patients has depression, and 1 in 3 patients has anxiety. For the autoimmunities, it is very important to look at this. Number one, it's a sign that this may be an autoimmune chronic spontaneous urticaria patient. Plus, number two, if you really diagnose de novo in a CSU patient Hashimoto, well, by all means, treat it, and you will have possibly double benefit. As for the consequences, cofactors, and components, well, coming back to the cause versus the components, the triggers, the cofactors that impact on disease activity in chronic spontaneous urticaria. Ask for them. Asking for it can sensitize patients that they may be relevant in them and can possibly be avoided, at least be recognized as something that makes sense to them. And finally, please continue to measure the course. This is important, not only in patients who you see for the first time. We should always, for the first visit as well as for consecutive visits, look at disease activity, impact, and control. What do the guidelines say? And they will say that in the latest version. We recommend that all patients are assessed for activity, impact, and control at every visit. And while the old guideline only had 5 tools to recommend, the 2 activity scores for urticaria and angioedema, the quality of life questionnaires, QQAL and IEQAL, and the urticaria control test, the guideline still says treat the disease until it is gone. And because we know that the disease is characterized by wheals, angioedema, or both, what we do in chronic spontaneous urticaria, and that is to use this algorithm to bring complete control to everyone, also means that we need to add one more tool that was missing so far, and that is the angioedema control test. You cannot, in patients who exclusively develop angioedema, no wheals, robustly measure disease control by the use of the UCT only. So we now have the complete list for assessing disease activity, quality of life impairment, and disease control in all patients with chronic spontaneous urticaria, those with wheals, those with angioedema, and those with both. And we are going to have tools for the chronic inducible urticaria patients as well. So be a pro, use the PROMs, the patient-reported outcome measures, to ask, is your disease under control? How active is your disease, and what does your disease do to you in your daily life? Now, I will be very quick here. This is really only a reminder. You all know the Urticaria control test. 4 questions, 5 answer options. The most point per answer is 4 times 4 questions, means 16-point maximum. And you want to get patients to 12 at least, to know that they are well controlled, that they're well controlling their disease. Now, what about this angioedema control test? Is it very different from the UCT? Well, it's very similar. Also has 4 questions with 5 answer options. 4 points maximum per answer option is 4 times 4. So the range is the same, from 0, no control, to 16, complete control. And here the cutoff is 10. And this tool, just like the other tools, has been validated for the use in India. Actually, for many of these tools, we have several language versions for India available. They're free to use, they're fun to use, patients like them, and they really help your approach to optimizing the treatment in patients with chronic urticaria. So I know that urticaria can be challenging. There's no question about that. It's a complex disease. We need to talk a lot to these patients, but let these tools help us to make our lives and the lives of patients easier, to bring to them what we really want to do, and that is to treat the disease until it is gone. In order to do that, we need 2 things. Know what's going on, and this is what we talked about today, and know what we need to do. And this will be the topic of the subsequent sessions of this series. And just to say, with what we have today, the treatments that are available, excellent antihistamines that can be updosed, omalizumab and cyclosporin, we can help most patients. We can't help all patients to the point where we want to help them treat the disease until it is gone. But most patients will see benefit from the use of the drugs and the algorithm. And for those who do not, it looks really, really good, because at no time in the history of mankind have we been doing more clinical trials in chronic spontaneous urticaria than today. These are all studies that are ongoing. These are all targets of modern emerging treatment options, targeting receptors on mast cells, mediators released by mast cells, cells that make mast cells become urticaria drivers. And I want to share with you just sneak preview 2 really interesting approaches, just in context, not in detail. And that is, we can today silence mast cells. Now, what do we do with antihistamines? We block 1 mediator, histamine, that comes out of mast cells. What do we do with omalizumab? We block 1 activator of mast cells, IgE. Silencing makes mast cells shut up and sit down, which in urticaria is a good thing. We can today also deplete mast cells in humans. No mast cells, no urticaria. So these 2 entirely novel approaches and strategies will and are being tested in chronic spontaneous urticaria, and hopefully make their way to us to treat those patients who do not benefit from the treatment options that we have today. But main point here is, let's do the diagnostic approach like it should be done, so we know what we're dealing with in each and every patient with urticaria, so that our treatment is targeted and can be improved by the use of the tools available. Big shout out to the UCARES in India and everywhere. A very rapidly growing network, more than 120 centers in more than 39 countries today, doing excellent work together. And let's also keep donating data to the Chronic Urticaria Registry. Very easy to find, www.urticaria-registry.com. Every physician can be a data donator. So let's do that. And please don't forget, we'll meet in December in Hiroshima for the UCare conference. So be there and check it out. It'll be the best chronic urticaria conference this year. And with that, greetings from dermatological allergology, the UCare here at Charité. I put my email address here on the bottom, so if you have questions that come to you tomorrow or next week, by all means, do reach out to me, and I'll be very much looking forward to questions and our discussion to follow. Thanks again for your attention, and thanks for having me with you.
Yeah, in fact, that was great, Marcus. As usual, you enthralled the audience. We are really interested to know more things. We have with us Dr. Krupa Shankar from Bangalore, Dr. Vijay Zhar from Nashik, and Dr. Abir Saraswat from Lucknow. So all the zones we are represented, we have experts. So we'll be having a panel discussion on the questions about urticaria, and then we'll take the audience question also to understand what they want from us. So with this, in fact, Marcus, you told us about the guidelines, which are the newer ones, and the thing. So what are the main difference between the newer guidelines? The basic investigation has been increased from 2 to 4. Like first it was CBC and CSR. So yeah, go ahead.
So there are a number of differences. First of all, I want to say that it's a much more global guideline than it used to be in the participation, and therefore also reflecting geographical specialties. That is important because it's a world guideline, and I really want to see the reach and the impact of this guideline grow. As for content, we have the 7 C's for the diagnostic workup on board. We have the angioedema control test as a new tool that makes it to chronic spontaneous urticaria. For the therapy, it's very interesting because we boil down the 4-step algorithm to a 3-step algorithm, which is really good because we need to make space for future treatments to come, like ligelizumab and others. And it really doesn't make much sense to talk about antihistamines as 2 different steps in the treatment. It's fluid. We want to optimize treatment with each and every class of drugs that we have by including information on how this can be done. And this is exactly what the new guideline does. We have first-line treatment with antihistamines, and we specify when and how to updose. We have second-line treatment with omalizumab, and we specify when and how to updose. And we have third-line treatment with cyclosporin. Again, we talk about the dose and what to do if it doesn't work. Now, this is good. This is cool because it helps us to bring patients faster to that goal. But what the guideline then also does is it provides guidance on what to do when you have complete response. So when to down dose or when to stop, what to do when you have partial response, how to optimize, and what to do when you have no response. So it's much more catered to the situations that we as urticaria treating physicians see in everyday life, and it allows space for other treatments to come on board with the next version of the guideline.
Yeah, thank you. That's great. Well, the panel discussion, the title is Challenges of Urticaria Diagnosis in India. So as you know, India is a huge country, more than 135 crores, and we are just 8 to 10,000 practicing dermatologists. So Dr. Abir, what do you think, what are the main mimickers of urticaria where patient might think that this is urticaria and this is not urticaria, which you have seen in your long practice?
Can sometimes be severe papular urticaria insect bite reactions in children specifically. Of course, urticarial vasculitis remains a mimic that's there in your textbooks as well. I have seen a few cases, maybe 2 or 3 of the autoinflammatory disorders. We published those as well. But those are really quite easy to spot in that the wheals don't respond at all to practically anything we usually do. And there are other symptoms as well, like deafness, amyloidosis, arthritis, things like that. So I think these are the, sometimes on superficial examination, erysipelas, etc., can look like urticaria, but I think within a half a minute you can make out the difference. But I think these are the main ones that we see.
Yeah, that's great. Vijay, anything else which is mimics urticaria? We know patients think that this is urticaria, but this is not urticaria.
Yeah, pretty well. This kind of arterial wheals, and sometimes the infectious disorders, like for example, in commonly what I see in clinical practice, like patients of scabies presenting with urticaria. The family of urticaria, sudden onset of urticaria in the family, and then you need to keep in mind such kind of infectious diseases.
Yeah, right. Krupa, any other mimicals? Do you think leprosy? In fact, many times my residents once diagnosed urticaria.
Yes,
and,
inflammatory.
Yeah.
I was about to say that early lesions of ENL.
Yeah.
Or type 1 reactions also.
ENL.
Yeah. ENL or even lepromatous leprosy, looking like urticaria, it does happen. So typically, this is more for all the doctors that urticaria, please confirm the diagnosis. Look for the wheals, should not last more than 24 hours. If they're lasting, please take the biopsy. That is the best option for this thing. And obviously, the challenges are many. In fact, we have an interesting presentation by Dr. Vijay Zawar on the difficult to diagnose urticaria after this panel discussion. So that will be taking this thing. Other things with COVID around, almost every day we get questions, doctor, can I take vaccine? I have got drug allergy, can I take vaccine? So Marcus, what is your take? It's very common. Every day I get 3 to 4 queries.
Every day, Kiran, every day. And look, and these patients are so different. No, some of them are very afraid of getting the vaccine. Others say, I need to be the first one to get the vaccine because I have this disease. No, so yeah, every day. Look, our general recommendation to all of our patients with chronic urticaria is get vaccinated. Why? Because we know from patients who have chronic spontaneous urticaria and contracted COVID-19 that this can be a really hard drive to high disease activity, long lasting. And this is confirmed by the global study that we did in the UCare network, where we found that 3 out of 10 patients with chronic urticaria who get COVID have exacerbation of their chronic urticaria. And in patients who are hospitalized because of COVID, it is 7 out of 10. So you need to see that against the very low rate. It does occur, no question, no, but the very low rate of transient mild exacerbation of chronic spontaneous urticaria that comes with vaccines. No, it is there. It is, no question, it is there, but it is usually a couple of days, easily controlled, or goes away by itself. And it is nothing compared to the risk that these patients are running when they don't get vaccinated. And they don't have an increased risk of hypersensitivity reactions. And we need to tell them, you are not allergic. You have no reason to think that your risk is higher than anyone else's. And the risk for an allergic reaction, of course, is super, super small. And that needs to be said again and again. When we say it with the societies, we say it with the patient organizations, we say it in the networks. We just need to bring that out there, because the same goes not just for chronic urticaria. No, there's the atopic dermatitis patients who are also asking me, and the mastocytosis patients who are asking me. You know, all of these patients with chronic inflammatory diseases that are afraid, because also of some miscommunication that is in the media, afraid of getting the vaccine, should not be afraid and should be encouraged to get vaccinated.
Yeah, that's right. That's the lesson, that all urticaria patient can safely take vaccine. Dr. Abir, what about drug allergy? Just to tell you one incident, one of our doctors refused to take vaccine because of drug allergy, and ultimately he got a severe COVID. So what is your take, Dr. Abir?
Drug allergies in patients who have who want to go for vaccination. I don't think drug allergies have an impact on this, because there used to be some vaccines earlier which would have tiny traces of antibiotics in them. I don't think these modern vaccines have any trace of any drugs in them. And I wanted to talk about another peculiar Indian problem, since we are discussing Indian patients here. Many of my patients across the board seem to think that the safer thing to do before getting vaccinated is to stop all your other medicines for whatever you're taking. Maybe it is urticaria or hypertension. stop them a couple of days before vaccine, then take the vaccine, keep them stopped for another week or so, and then restart. So this is disaster for chronic urticaria.
Exactly.
So we have to tell them against this as well. And this is the predominant instinct somehow in Indian patients. They're not so scared of the vaccine. They just say, let me stop all the other things, then I'll take this, and then I'll restart after some time.
Yeah, so that's important lesson. Please do not stop any medicine. Continue all the medicine and take the vaccine. And because vaccine, there is no contraindication. Either you have got a drug allergy or urticaria. Dr. Vijay, anybody has got a food allergy? What do you tell them about?
Generally, food allergy is not a big issue in chronic spontaneous urticarias as such. It is much less common than it is believed. Actually, patients believe that they are allergic to something, but in clinical practice, the risk is much low.
Yeah, right. That is important. Food allergy is not the true allergy in urticaria. So, Marcus, somebody having food allergy and they say that, doctor, I will not take urticarial vaccine because the COVID vaccine because it might contain egg and fish. How true it is?
To me, there's no problem.
No
Food
problem.
allergy
Exactly.
is not a risk factor for allergy to the vaccine. Look, these cases are so super rare that they're individually published. Like an IgE mediated hypersensitivity to PEG or unless you had previously had a severe allergic reaction to another vaccine, there is no risk factor that makes it more dangerous for you to get the vaccine. And even in these patients who are super rare, we can do testing before we vaccinate them. And when the test comes out negative, we still recommend to them that they get vaccinated, maybe in different settings, with a little bit more response in place. But look, here our NIH says everyone who doesn't get vaccinated will get COVID-19, period. The whole thing with herd immunity and whatnot, it's just risk, okay? We will see these new variants, and everyone who does not get vaccinated at one point in their life will get COVID-19. You don't want COVID-19 if you're a urticaria patient. I mean, no one wants COVID-19, but if you're a urticaria patient, you really don't want COVID-19, So.
Yes, that's right. That's the thing. Dr. Krupa, somebody has got true anaphylaxis. In fact, that is actually true. Somebody who had a history of anaphylaxis, you have to be extra careful while taking vaccine. So what do you tell about these patients?
Keep them for a longer period of observation, that's all. And the thing is, if the anaphylactogen, the substance which is inducing anaphylaxis, has been recognized, then there is no problem at all. Like as was just mentioned, if there has been a reaction, severe one to a previous vaccine, should you be concerned about the present one? Otherwise, no. Anaphylaxis is generally diagnosed by history. So most people who have an anaphylaxis and have consulted a doctor will have a general idea of what's likely to precipitate it. If it has nothing to do with an injection at any point in time, go ahead with the vaccine.
Yeah, right. Obviously, little extra care is required. Otherwise, no. Well, this pandemic is going to stay and going to go ahead. So actually, Dr. Abir, what is your take on the COVID, the pandemic and the urticaria patient treatment? Do they come? What is happening to most of our urticaria patients?
A few patients who got symptomatic COVID and their urticaria worsened. But in the vast majority, in my case, it was because they had stopped their medications. But of course, some of them may have continued also. And this is well known, exacerbations do occur. An interesting thing I found out was many of these patients had dramatic remissions of urticaria, even when they did not updose their antihistamines in any way. But this was purely because of so much steroids being pumped into them as part of the COVID strategy that was so prevalent in India. GPs were giving steroids to even mild COVID patients. So many patients said, I was wonderful for 15 days or 20 days after COVID, but now your medicines are so, they don't work as well as my COVID medication did. So we have to tell them that you were getting steroids, and you can't do that for a long period of time. So this is again something peculiar that I heard from patients during the second wave.
Right. That is important.
Can I add something?
Yeah, Vijay, please go ahead.
I have seen some of the patients getting exacerbation Initially, I thought if it is because of vaccine or COVID infection itself, but patients keep on taking OTC, NSAIDs and other drugs, during this pandemic. Sometimes hydroxychloroquine. I think a partly the drug-induced urticaria triggers are known in this pandemic.
Right, because everything is possible in India. Patient can go and buy medicines and take, and that is what used to happen. In fact, there was a whole lot of COVID treatment on the floating on the WhatsApp, and people used to follow. Krupa, what is your take? What out of our urticaria patients who
every
are
single
on?
COVID patient, every single COVID patient in India was getting doxycycline, azathioprine, azithromycin, and 3-4 more things.
Cocktails, yeah.
Multivitamins.
True. That was a very sad thing to happen, but it happened in India. This thing. Krupa, what about the patients on immunosuppressants and urticaria? Anything you face problem during COVID?
No, I mean, there are published guidelines which you can easily follow. With any of the immunosuppressants that we commonly use, the risk is only slightly high. We need to be considered only if we are dealing with a live attenuated vaccine or something like this. Otherwise, not a problem.
Yeah, right. Marcus, biggest problem what many patients face, and those who are on cyclosporine and they do take vaccine and methotrexate, because these two things are commonly used in India for urticaria. Although there are guidelines, but what is your personal message to all the dermatologists in India about cyclosporine and urticaria and methotrexate?
Look, I think we ought to live by the mantra of the guideline: no, treat the disease until it is gone. And that's easy to say in a country like mine, where I can prescribe omalizumab essentially to everyone I want to. No, there's no limitation to the number or quality of urticaria that I can treat. And I do know that this is not the same everywhere on this planet. So we have to keep these disparities in mind when we make these recommendations. And the recommendations are always meant to have that little side note that says where available. No, use an antihistamine where available. Updose where available. Use omalizumab where available. No, not everything is available everywhere. So that's the one thing. I used to use a lot of cyclosporine, Kiran, a lot. I remember days when I had 60, 70 patients at the same time on cyclosporine, and I never do a long cyclosporine or did long cyclosporine. No, I get antsy after 3 months, and after 4 months I get really nervous, and after 5 months I stop. No, even if the patient is doing fine side effect wise. But I haven't used it last year. I haven't used it the year before, because we can use omalizumab and it usually works, even though in those patients who have autoimmune chronic spontaneous urticaria, it may take 3 months or 4 months or 5 months before they do respond. Would I use it? Yes, I would use it, because you need to control this disease, and I would use it even in the times of a pandemic, where generally you say immunosuppression in autoimmune diseases or for other reasons should be done with caution. But that's exactly it. Let's do it with caution, and then we will get the best outcome for the most patients. It's for sure better than not treating. No, but my third point: when you can give omalizumab, then you should do that rather than cyclosporine. No, it's the longer acting, better, safer, more effective drug.
Right. So if you can afford, omalizumab is the best answer. It's not immunosuppressant. The patient can safely take it. Marcus, I was just talking about the new guidelines, and I got a very good remark from some dermatologist. He said that 10 years back, you used to say updose antihistamines. Now this time you are saying you updose omalizumab. So how true it is? What are your new guidelines recommend? How much you can updose omalizumab?
Oh, yeah, sure. No secrets here. So we still say for the antihistamines up to 4 times. Don't do it too long, because whatever doesn't happen after 2-3 weeks will not happen later on. For omalizumab, we say you can go up to 600 milligrams every 2 weeks, which is also the highest that I do. I have to say that the best thing that you can do to an omalizumab partial responder is indeed to give more. But I see that when I go from 300 to 450, that's when I make a big jump. Or from every 4 weeks to every 2 weeks, this is when I see that, well, another half of my patients get true benefit. At the end of the scale, no, when you're already at 600 every 4 weeks and you say, can I get a better response? Then it does happen, but it's not as likely that when you give 600 every 2 weeks as it is from going from 300 to 450 or from every 4 weeks to every 2 weeks. No? So still try it as best you can. 600 every 2 weeks is safe. You know, this is a dose that we've been using in asthma for a long time. So there's no risk there really, and it can work, and it should be tried.
Right. So that's the thing about the thing. We have many interesting study this year from UK and from this thing. We'll come back to it. This thing. In fact, there are many questions as usual. You know, the urticaria patients have got more questions than the answers. In fact, there is one question which is being asked is should IgE be performed in every patient? What is your take? IgE. You have mentioned about the IgE and IgG anti-TPO. So all the dermatologists should use it regularly?
Yeah, that's my recommendation. We are specialists, no? So these patients have already gone through GPs, at least in my country, and usually more than one, plus their grandmother and the pharmacist and the neighbor and who not, no? So now they need someone who knows what they're doing, like us, no? And in order for us to know what we're doing, we need to ask the right questions and do the right tests. And if you want to know, is this a patient with autoimmune versus autoallergy, which to me is very important to manage expectations and for other things. IgE, I don't know, Kiran, how much is IgE in India to do? Here it's not pennies, but it's not expensive, no? And IgG anti-TPO is the same thing. So it is, from my point of view, worth the investment of doing these tests. And yeah, I think that at least if you're in a hospital-based setting or you have a good network and a lab to work with, these tests are widely available, no? And the money that it costs to do these tests is worth spending it.
Right. So IgE and anti-TPO, it is easily available in India, and we as a dermatologist can do it because low IgE and high anti-TPO points to the autoimmunity. So that is one thing. I just had Marcus query. You said 10% is autoimmunity. Well, 1/3 is inducible urticaria. So how much is autoallergic according to you? Because that we don't have any rough estimates.
Kiran, if I had an answer to that question, I would sing it from the rooftops, but I don't have that. And it is because we're still working on the commercialization of tests for autoallergy. Look, what we can do right now in very specialized settings, all of them UCARES, by the way, is we can measure IgE anti-TPO. Our friends in Colombia can do that, in Korea can do it, in Italy can do it, we here in Berlin, we can do it. But this is not something that you can buy from a company, no, or order at your hospital lab. This is something that's really restricted still. And even if you have someone who's IgE anti-TPO positive, which I think then makes them autoallergic, well, they could be autoallergic against something else. For example, interleukin 24. We know that 60% of patients with chronic spontaneous urticaria have IgE to interleukin 24. So you can ask that question and the answer you will get, well, 50% are TPO allergic, 60% are interleukin 24 allergic. And you know, there's a number, 5, 6, 7 autoallergens to which we now can measure these IgEs, and there will be overlap where one patient is allergic to more than one autoantigen. That's the autoallergy group. From my point of view, to make this a little bit more palpable, two-thirds of all patients have autoallergic urticaria. What about the other third? Well, we found that only 10%, 8% to be precise, have true autoimmune chronic spontaneous urticaria. But if you remember that Venn diagram, there are plenty of patients who are positive for 1 or 2, but not all 3 of these markers. What's up with that? From my point of view, the basophil activation test is probably the best single individual test to diagnose chronic spontaneous urticaria due to autoimmunity, and it is positive in 30–40% of patients. So my easy look at this, and I hope that the data that will be coming out will support this, is that 1/3 have autoimmune chronic spontaneous urticaria, 2/3 have autoallergic chronic spontaneous urticaria. There will be a little bit of overlap. We will find patients who have both. We may even find patients who first have autoallergic and then become autoimmune. And there will be patients who have neither autoimmune nor autoallergic chronic spontaneous urticaria, but I think there'll be very few. That's where we stand today, and we really need to, as a community and as a network, bring these tests to a broad number of centers and patients to learn about the prevalence and what's going on. No?
Yeah, thank you very much. Abir, we have Bilastine tablets available in India. Has it changed your practice, and how powerful and how good it is?
It hasn't exactly changed my practice, but I have been using it. I have been prescribing it to patients, and it seems to be effective. More so, I think maybe it's because we get more severe patients, but once a day dosing doesn't seem to do a great job in quite a few patients. So my standard starting dose is BD. But then if I look at retrospectively my other prescriptions, even other antihistamines, we as specialists tend to start at slightly higher doses anyway. So that was my impression about Bilastine. It seems to the other best non-sedating antihistamine that we had earlier, which was fexofenadine. There's a subset of patients of mine, everybody has them, who get very sedated with fexofenadine. I shifted a few of them, or almost tried to shift all of them to Bilastine, and almost half of them did not have sedation. So that's a good solution that I found for that group of patients. So that's my overall impression so far.
Right. And as we know, we are all dermatologists, so we are going to get the severe urticaria patient, not the routine. So typically, most of the time, we need to give double dose to most of our patients. And as I agree with this thing, fexofenadine and Bilastine, these are two things which are truly non-sedating. And Bilastine scores, as you have said. Dr. Vijay, what is your take on Bilastine tablet, and how do you put it in your practice now?
Generally, most chunk of our patients are fungal infections and the other pruritic disorders, apart from urticaria. So what the literature suggests is there are minimal drug interactions of Bilastine. So I think we can little more confidently can use updosing of Bilastine.
Right. So this is one of safe drug. Krupa, what is your take? You give one tablet twice a day and then 2 tablets twice a day, or how do you go about? Or you can give evening, you can give something else?
No, it is generally that way. One tablet twice a day, up to 2 tablets twice a day, but then it takes some time to kick in. It takes about 3–4 days for it to gain some wheel control, sometimes up to a week. So if we are going to give Bilastine, or when I give Bilastine to my patients, I tell them that this will take some time, please be patient. If by 1 week they have not achieved complete wheel control, it's either for time for updosing or change of antihistamine. That's the point at which I take a decision. If it's going to work, it generally begin on the 4th day or 5th day, something like this.
Yeah. Marcus, you have been using Bilastine for a long. We have it since about a year or so. So they mention about you take it on empty stomach, and many times patients forget to take on empty stomach. So how much really it's relevant?
Nothing. I don't lose my time talking about this. If I recall correctly, it's like 15% or something. So urticaria patients need a lot of time, need a lot of my time, no? And there's never enough time with the urticaria patients. So I'd rather focus on things that are relevant, and taking it on an empty stomach or what is not part of what I talk about. Not relevant.
Right. So don't worry about this thing empty stomach. You can start in after 2 or 3 days. Obviously, you will get the sustained levels and good efficacy. Marcus, I would like to take your attention to the very important paper you just released about the depression and urticaria, which you have shown that substance P levels are important. Can you just highlight us, because that is one of the best paper in this year which I saw.
Thank you. Well, that's really cool. I should say that this was really driven by the UCARE in Istanbul with Emek Kocaturk, and a very nice collaboration between our 2 UCAREs. Look, we knew before that substance P is linked to urticaria because substance P is a neuropeptide. No, it's released by sensory nerves when people are stressed. It binds to several receptors. I'll come to that in a minute. So we knew that levels of substance P are elevated in patients with chronic urticaria, and we also knew that disease activity is correlated, not tremendously strong, but statistically significantly correlated to disease activity in chronic spontaneous urticaria. Now, why is that interesting? Because it turns out substance P is one of the main ligands to the mast-related gene-protein coupled receptor X2. Wow. Big name, big receptor. Why? Because it has many ligands. It is on mast cells. It is upregulated in the mast cells of chronic spontaneous urticaria patients. And it is a wonderful mast cell degranulating receptor. Now, you put these two things together, you have substance P, which is elevated. You have more mast-related gene protein coupled receptor X2 on mast cells. Boom, you have the perfect combination for disaster. Now, what was interesting about this paper was that we found that there's a link to both, no, of the substance P levels to the urticaria and of the substance P levels to the comorbid depression. Now, depression has also been speculated to be linked to substance P. So it may be that we finally figure out how urticaria and depression are linked by one molecular mechanism, and that is substance P, elevated substance P. You know, this idea, well, it's more of a concept that chronic inflammation results in signals that cause depression. That has been around for a while, but it has been very difficult to link it to cellular and molecular mechanisms. And yeah, while substance P has been a focus of these speculations, it's not really easy to find hard evidence on that. And our paper may be one of the first to really connect these dots and to maybe start to make sense of higher or elevated substance P levels, overexpression of mast-related gene protein coupled receptor X2 in a mast cell-driven disease with a high rate of comorbid depression. So this is probably the merit that describes this paper.
Yeah. So first time there is the evidence and the proof to show that depression and urticaria are correlated. Maybe more papers will make it very clear, but congratulations for that nice paper. Dr. Abir, urticaria patients are always there with you, and obviously you must be getting toughest of tough patients. So when your patients with 4 tablets not responding, you will start cyclosporine or omalizumab. But anything else you try apart from omalizumab and cyclosporine, those who can't afford omalizumab?
I always give montelukast also to most of my patients. Usually when I'm up dosing to 4-fold, I'm usually adding montelukast as well, trying to save some time, or a fairly innocuous drug, cheap,
Yes.
not many side effects. So I usually do that. Some patients who have symptoms of dyspepsia or GI symptoms, I do, or sometimes even blindly, I sometimes try ranitidine as well in these scenarios. And of course, if patients who have slightly longer lasting wheals, maybe not to urticarial vasculitis levels, but some patients are there who have wheals that last 8, 10, 12 hours, then I also take this as a signal to try Dapsone. And sometimes it works. So these are some of the things that I try.
Yes. So remember, Abir rightly said, you give 4 tablets of antihistaminics, you can give 2 tablets of ranitidine and 1 tablet of montelukast, and then think of Dapsone and then methotrexate, cyclosporine as per patient's pocket, because everybody can't have omalizumab. Dr. Vijay, what is you updose 4 times, then what is your next step of action? What you do next?
Generally, the second line of drug that you have thought of, most of my patients are not affording for omalizumab or the other drugs. Cyclosporine is a reserved drug. Some of the patients whom can be taken for little tide or crisis for urticarial patients, I use cyclosporine. But other drug that often I use is methotrexate.
Okay. Can you tell me how much methotrexate you use? How long?
I generally start with 7.5 to 10 milligram a week, and then I can step up to 15 to 25 milligram. But that gives good result. Only it is slowly acting, but we have much more experience of using methotrexate in psoriatic patients. So I feel little more comfortable in using methotrexate in place of other drugs as compared.
Right, patient. Yeah, Marcus.
Kiran, is omalizumab available as a biosimilar in India, or?
Yeah.
Okay, and
No,
it is
we
paid
have
out
got
of.
biosimilar.
Okay, and it is paid out of pocket. All patients have to pay for it themselves.
Yes.
Yeah. Most of them, except less than central government and this thing, but otherwise it's to pay from pocket. But now we have got a original molecule and a biosimilar also available. So biosimilar is little economical, whereas the original molecule is little costly. But still, it is beyond reach of everybody. But very few people get it from the reimbursement. But otherwise, it is from the patient's pocket.
Okay.
That's the thing. But slowly it will improve because the biosimilar cost is coming down, and it does work. Obviously, not like original. Yeah, but that's the situation here. Krupa, what is your take when you give 4 tablets of antihistaminic? What you start after this thing?
Yeah, same as what Abir just said, or you mentioned earlier. Ranitidine, Montelukast. Dapsone, I don't reach for it very early unless I see a lot of neutrophils in the biopsy, because inevitably when you give Dapsone to a non-Hansen's patient, that is the one who will get Dapsone syndrome. So I'm very cautious with Dapsone. And if I find lots of neutrophils, and if I'm hesitant to give Dapsone, I use colchicine instead. That works in some of my patients. So that's the way. I mean, if but then omalizumab and cyclosporine are already there, we'll have to reach for one of those inevitably. By and far.
Right. Thank you. We have got last 10 minutes or so left. We are getting many audience questions, so we'll be taking that and then this thing. So typically now there is one question by Dr. Himanshu. He's taking Bilastine. You are mentioning about 1 tablet twice a day or this thing. Is it officially approved for doing updosing in India or abroad? Abir, your take? It's not approved for double dosing, right, in India?
Not yet.
I'm sure everywhere these are off-label uses if we increase the dose twofold. I'm not sure about Europe, but here, of course, authorities don't give approvals for higher doses. Those are left to the discretion of the treating doctor.
Yeah. So it is safe to updose. Bilastine that way has got not many drug-drug interaction. In fact, that is the advantage of Bilastine over other molecules. This thing. Yeah, there is one more question, Dr. Marcus, for you. Can interleukin 24 levels be a marker for CSU, chronic spontaneous urticaria?
They may be, but I think that before we measure interleukin 24, we should measure the IgE to it. That would be much more relevant because that is what drives the mast cell activation. There is a group who has shown very nicely that interleukin 24 is increased, especially in the lesions so in the wheals of patients with chronic spontaneous urticaria. So then you have the IgE, you have elevated levels of the autoantigen. Again, perfect combination for disaster in the skin driving whealing.
Right. So obviously they're all advanced interleukin levels to check, but obviously these are to be done in the advanced center, and well, IL-24 have a feature, so you might get it in the thing. Right. Yeah, Dr. Marcus, your talk was more on the advances in diagnosis, but I would like to know what are advances in treatment. What you see in next 2-3 years, what will come, which will be easily available, which will be suitable. You said about fenibrutinib and legelizumab. Which are your best things which can be useful? We'd like to know from your.
Look, I'm excited about every new trial that comes to the urticaria community. The soonest one to come to us for sure will be legelizumab. No, it has finished the phase 3 trials. It is better than omalizumab. So here you go, another anti-IgE. We've also seen very exciting data from Bruton's tyrosine kinase inhibitor. Fenibrutinib and remibrutinib is also being studied right now. That's an interesting concept, no? Because it blocks mast cells downstream of the IgE receptor, and it blocks B cells and therefore autoantibody production. It's an oral medication that is becoming, with every new generation of BTK inhibitors, safer and safer. So I'm quite excited about that. But I don't want to stop there, because we're looking at anti-IL-4 receptor. Very cool. Anti-IL-5 to take out the eosinophil game. All of this has been shown to work. together with Elias Toby and the UCARE in Haifa, we gave secukinumab, anti-IL-17, to patients who failed omalizumab. 8 out of 8 responded. So I think, you know, if it's an autoimmune disease, we ought to treat it like an autoimmune disease with anti-autoimmune drugs. That's a good start, no? I'm quite excited about complement 5A, because we know from the work that Alan Kaplan and the British team did that C5a is a co-activator of mast cells when these autoantibodies go there. And there are 2-3 different anti-C5a drugs that are available today that could and should be tested. But we also have these really cool new things that are being tried only in urticaria, no, or mostly in urticaria. Silencing, and lirentelimab is the antibody that so far does it best. It goes to Siglec-8, which is an inhibitory receptor on mast cells. And this is the most radical idea. I'll talk about it at EAACI this year. We're looking at patients with mast cell-driven disease. No mast cells, no disease. If we deplete mast cells, no, this thing must stop. And I will share data that is just very, very interesting from chronic inducible urticaria, but this must also be true for chronic spontaneous urticaria. We can do it with an anti-KIT antibody. You know, mast cells need KIT to detect SCF. Without SCF, they die. No? So if you use that antibody, you starve them for the SCF and they go away. And that is a very, I think, very convincing mechanism to use to treat patients with mast cell-driven diseases, including, but not limited to, urticaria.
Well, that is true.
Kiran,
Vijay.
We have Abir with you, and Dr. Abir has got a very good experience of secukinumab in the clinical practice, so we can invite his opinion also.
Yeah. In fact, when Marcus said secukinumab, I could see a smile on Abir's face, and I said, well, in fact, we do use secukinumab. Abir, what is your take?
I haven't tried it in few enough psoriasis patients that are able to afford it. I can't do these kind of trials. You know, if they don't respond, my patients are going to be off with my head. So I can't try this because most of them are paying out of pocket. But it's interesting because IL-17, another disease where IL-17 is supposed to play a central role, is vitiligo, and secukinumab has been tried there as well, although the initial results were not as good. But so basically no personal experience. But it's interesting, very interesting.
Yeah, interesting. In fact, we tried in secukinumab even in hidradenitis suppurativa, but obviously the results were not as great as this thing. So obviously newer discovery, newer changes, newer mind, and you can think and something out of box can happen, and that could be a serendipity or some new drug. But well,
I can share one patient. I had a patient with severe urticaria and psoriasis. This patient was insured and he was hospitalized for approved use of secukinumab, and his chronic urticaria dramatically responded. So it is in the line with what Dr. Marcus has said.
Right. We have got last 5 minutes to go this thing. Yeah, there is one question, Dr. Abir, apremilast in CSU. Any of your take? Have you tried?
No, I would love to. I haven't tried it yet. I'm not sure any of you colleagues know. I think we're being negligent by not going into urticaria with these novel drugs. Although we don't understand much about how mast cells relate to these targets, we ought to try it. You know, those are good drugs. Safe by and large, and urticaria can be devastating. So what's the harm? You know, we need to really explore every option that comes to us.
Yeah. Any Krupa, have you tried this apremilast in CSU or anybody?
No.
Well, there's a question from Dr. Jain.
The one I can come closest to is apremilast being very useful in atopic dermatitis. Urticaria, no.
Right. So, well, that was a thing. This thing, role of azathioprine in CSU. Again, one question by Dr. Abir. Any experience on azathioprine? Dr. Amit is asking this question.
No personal experience. I know some colleagues have tried, but I haven't.
I have read some of the reports where some of the investigators have used azathioprine in place of other immunosuppressants.
Yeah.
But azathioprine takes a long time for the onset of action. Secondly, it's not my favorite drug simply because it is unpredictable with the bone marrow depression as major side effect. So I'm little scared of it. And some of the patients, you can do the TPMT levels, but these are again more expensive than the drug itself in India.
Right. So obviously, azathioprine is not the routine. This thing preferred one is
on T cell, not B cells.
Yeah. Say again, Abir.
Azathioprine preferentially suppresses T cell induced immunity, does not have as great an effect on B cell. So if you want to somehow hit the B cells, cyclophosphamide would make more sense, but that's again a drug not to be taken lightly, and
yes,
I don't really like to use it.
Yeah. Right. So obviously, message is clear. The preferred drugs are cyclosporine, omalizumab, and methotrexate to some extent, but others are less oftenly used. Well, last few things about autoserum therapy. Vijay, you have also published autoserum therapy. What is your take and how do you put it in India? Because every year we see publications from India and from the few countries.
Yeah, to put Marcus' words in the combination of this study, I can say that this is poor man's biological for my patients.
Yeah.
It does work in some of the patients, but it is unpredictable in which patient it will be dramatically effective. I have some of the dramatic experience with anti-serum therapy working well, but it is, like other drugs, we can't predict which patients will do really well.
Yeah, that is true. In fact, Abir did the first study, and A.K. Bajaj sir still used to tell me that patients to come with him with the syringe needle with his hand. Hey, doctor, somebody got cured with your autoserum therapy, so give it to me also. So Abir, you have got those fond memories of that first trial?
Very wonderful paper, Abir. Old wine in new bottles.
Something like that.
Very nice paper.
I did a lot of study. I did a lot of patients during that time. And again, what Dr. Vijay said, it's completely unpredictable. We tried to correlate it with ASST, etc., IgE levels. Didn't make sense. And some patients responded dramatically, as Dr. Vijay says. Some patients don't respond much at all. The placebo effect is very, very strong because you're taking out blood and injecting. And, for patients who believe that it will work, I think it's a powerful placebo effect as well. But yes, there are certainly dramatic remissions which you cannot ignore in say about 10 to 20% of patients.
Yeah, Marcus, anything new on autoserum therapy?
Well, you know our papers. You know that I am at heart an autohemotherapy fan. I get a lot of heat from my German colleagues because they think it's voodoo. I think it is unexplained immunotherapy. I would love to figure out the mechanisms. I've seen very good results. And I also, Abir, I worked with the hypothesis that this would be better in ASST positive patients. It is not, in my experience. I shot that theory in the wind. The last finding was that it is linked to IgE, anti-IL-24. So maybe with these new concepts that become available and tests to test for them, we will figure out who the responding subgroup is. If you work with the idea that there are at least two subgroups, then it makes sense that some patients don't show any response and others show a good response. I love the idea of curing urticaria before I retire. I'm not sure that will happen, but I think we ought to keep working for cure. No, we need to figure out how to make this disease stop, not just the symptoms. And I think with the idea that you can be allergic to yourself and therefore maybe also benefit from specific immunotherapy, which cures allergy patients. So why not have that vision, have that dream that you can make people who became allergic to themselves tolerant to themselves again? And maybe, pure speculation, maybe this is what we're doing with autohemotherapy. Maybe we're taking the allergen, the autoallergen, and maybe we're exposing it in a way that is used by the immune system to generate a tolerizing, retolerizing response. I think we shouldn't give up. No, it's too, clinically, it's too effective to just be placebo. And from an immunological point of view, it's too interesting to say it's voodoo. We need to figure out how it works. And then we will also figure out for whom it will be of benefit and why.
Yes, yes, yes. So obviously, people more research is needed, but there are therapies which people still manage, and it can be used in Indian setting. And Indian patient love injection, as Abir has said. Well, we are almost at the end of our this thing. In fact, I just want last two lines from all of you about urticaria markers. So then we got interesting presentation by Dr. Vijay Zawar on the difficult to treat cases. So Dr. Marcus, just last few lines for Indian dermatologists for urticaria.
Look, I'm very happy with the progress that we're making. I'm so happy about the strong urticariology in India. You guys are a true force with the network, but also globally. I love how much insight comes from India, but also how many challenges come from India. No, we should not become complacent with this tremendous progress that we've made over the years in the understanding, in the new trials. We need to push each other, and that's what you do, and you contribute to us moving forward. On a clinical point of view, we started with the delay in diagnosis, and this is something that I had underestimated in my country a lot. I thought it is I still think it is very easy to diagnose chronic urticaria, but apparently non-specialist physicians are troubled by this or struggle with this. So we ought to spend at least some of our energy and time in educating less specialized physicians who are inevitably the first physicians to see urticaria. It is not us as specialists who see patients first. They need to be able to diagnose early. They need to be able to go at least the first step of the guideline recommended treatment. And I think we need to help our non-specialist friends with that by education, by telling them, let your patients show you their cell phone with the wheels on them. It'll make you so much more confident that this is chronic urticaria. Ask the right questions, do the right tests. I think this is possible for all GPs, for all pediatricians to learn. And with chronic urticaria being so prevalent, no, 1 in 100 people on this planet today has chronic urticaria. We need to educate many more doctors much better in order to help all of these patients. And thanks for doing that.
Yes, yes. In fact, the UCare handbook of urticaria is in the process, which will come soon and which will help many of the GPs, pediatricians, and non-dermatologists to this thing. Well, thank you for all the support. And well, we'll be getting a handbook for physicians, and it is written by all UCare authors all over the world. Yeah, that's all. Dr. Krupa, any last two things about urticaria? Anything you want to tell to audience and the dermatologists?
Well, I think everything, all bases have been covered. We need to know that even now there are people who believe that urticaria is secondary to something else, and they still spend a lot of time and money trying to find a cause. If anything has come out of the urticaria guidelines over the years, it is an understanding that urticaria should be in the face of attempting to discover something and not finding any underlying cause. It's a disease in its own right. It has its own salience that we need to recognize. If we wrap our head around that particular concept, then the patient will be much better managed. If you spend all your time and resources searching for an underlying cause, that will waste the patient's time, your time, and the patient's resources, and will frustrate you. So the salience of urticaria as a disease in its own right is point number one. Once we recognize that, how we deal with it, of course, there are people to guide us, there are guidelines.
Yeah, thank you. Dr. Vijay, last two lines, anything you want to tell to dermatologists?
I think most of the things are covered. So
yeah, thank you. Yeah. Yes, thank you. Abir, anything want to tell about this thing? Otherwise, in fact, we had a very good discussion. We almost took 50 minutes on this, and it was really interesting. And all Marcus made a difference. He made a difference. He told us about so many things, and we are delighted and excited. Thank you, Dr. Marcus, Dr. Krupa, Dr. Vijay, and Dr. Abir for this interesting discussion. I'm sure this will be a treasure for all of us to remember and take it forward. And obviously, we as UCares do work, and please, all of you can join UCare centers. The UCare website is there. In fact, we are happy to help for you for anything you want for urticaria. With this, I wish to conclude this question and answer session. And now I request Dr. Vijay Zawar to come forward and show his presentation about the difficult to treat, difficult to diagnose urticaria. Thank you very much. Please stay tuned for the presentation.
How can I share?
Yeah, share slide from the share screen. Yeah, we can see it. Vijay, we can see your presentation.
Am I audible? Yeah.
Yes, go ahead.
Okay. At the outset, I must thank Marcus and Kiran for allowing and encouraging me to present on this platform. Thank you so much. Here is a small presentation actually, is not actually about the difficult to treat, but some of the striking and unique examples of chronic urticaria patients, or not necessarily even chronic, acute on chronic, or some of the acute exacerbations of urticaria, as I notice in my clinical practice. So, as you all know, that triggering of urticaria by infectious agents is common thing in the acute urticarial patients. However, its role in chronic urticaria is quite uncertain. It's not clear. And in the spontaneous acute urticaria, there is no doubt that there is causal relationship. But in chronic urticaria, uncertain. But at the same time, there is a concept. I think there are a couple of wonderful papers on this subject from the European authors. And one of the author mentioned that whatever is a chronic urticaria because of infectious, it has started as acute after the infectious injury. And I think this is a bottom line. Some of the presentations, some of the cases that I have seen is these kind of striking examples. These are, of course, they are not the exclusive, but they are exceptional. So there are very few studies with the correlation. So my first patient, 18 years old male patient, he suffered from chronic urticaria for 1 year. There was no history, not related to drug or anything systemically going wrong or fever or something like that. Battery of investigations as usual was negative. And he used to get quite recurrent exacerbations noted with the recurrent episodes on the body. And the cause of flares were unexplained. He used to have baseline chronic urticaria. Sometimes the activity of the urticaria used to wane and wax, but the flares, kindly forgive for the spelling mistake, flares remained unexplained. He was treated with various non-sedative second generation antihistamines, which at times required updosing. He received few shots of steroids even from general practitioners. And there was good control with this, but he used to immediately relapse after the withdrawal of antihistamines. Now, sometimes the value of careful clinical notes, sometimes you got to listen to the patients, and sometimes you need to be a little bit more observant. It was noticed that during clinical examination, he often used to get acute exacerbation noted, and the disease began in the groins as well as the lower abdomen to begin with. That was the anatomical area. So he had, history of tinea cruris for 3 years, where he used to treat it with the self-medication, often over the counter, with the combos containing potent topical steroids. And he had this kind of, tinea incognito, which definitely means that it is unrecognized tinea because it is not presenting in the classical forms due to application of chronic steroids. And his KOH and culture were positive for T. rubrum. See, now this patient, along with the antihistamines and updosing and all, I just added itraconazole therapy. And within 2 weeks, his urticaria became, very inactive. At the same time, the non-sedative antihistamines were gradually withdrawn, and I could completely stop the disease of, tinea cruris was further taken over for the topical sertaconazole treatment. He did fantastic. So recurrence of urticaria in this patient actually stopped for 1 year. And so the final diagnosis was tinea incognito presenting as chronic urticaria. So urticaria associated with dermatophytosis is known, but not widely published, not widely known concept. The potent topical steroids used for treating tinea infections in India has caused havoc. It has caused an epidemic proportions of resistant tinea coming up with varied presentations. And that may be, speculated because of altered immunological responses to the inflammatory process in the tinea infections due to chronic steroid applications. This is said to be responsible, but we need to, do more studies and prove it beyond doubt. This was our old paper, a decade ago. We observed this even before the current epidemic of resistant tinea came in. So my another patient, an otherwise healthy male adult, suffered from chronic spontaneous urticaria for 2 years, not related to any obvious drug or any other systemic disease, no background of allergy in the past of the family. He also had the intermittent flares, which remained unexplained. He was well controlled by the double dosing of the non-sedative antihistamines, but sometimes it used to go out of control, and I used to feel like whether the disease is not getting adequate control because of only antihistamines. A careful history in this patient, again, it was noticed that during the clinical examination, he often used to get acute exacerbations noted, which started on as an irritation over the genitalia. This patient was very specific. I used to ignore his observation, but carefully after looking at, I told him, you come repeatedly and show me the initial exacerbation. He used to, even predict what irritation will lead to now. Or irritation over genitalia and profuse crops of the arterial wheels all across the body. So he had history of promiscuous sexual behavior and was treated in past for genital herpes. His Zhang smear was positive, even herpes antibody was positive, and it was proved beyond doubt the genital part of it. But I was not sure whether this could be linked. But to my surprise, this patient, after starting acyclovir therapy, 200 milligram 5-hourly for 7 days, and then continuing the non-sedative antihistamines, which were gradually withdrawn, and all the recurrences in this patient stopped. So it was, a fantastic response like to antiviral therapy, which was my retrospective observation probably. And so the final diagnosis was whether herpes simplex virus is associated with chronic urticaria in this patient. It is said, it is published quite long back in 1998, upregulation of cytokines with the acute phase response leading to temporary state of enhanced mast cell releasability is probably the mechanism for aggravation of urticaria during the viral infection leading to chronic urticaria. And our observation was published in the international journal some time back. So thank you, Kiran, for helping me to write this paper. My next patient was actually not chronic urticaria, but this was a patient, lady, with recurrent unilateral urticaria on the face. And I was wondering whether it is because of sunlight, whether it is because of chronic application or what not, or whether it is a cause of drug reaction or something. But nothing, was traceable in the history. So I just tried to observe each episode. I observed 3 episodes of this lady, and there was a complete resolution after treating this patient for, she had acute inflammatory reactions on the upper lips, and which was proved to be because of herpes simplex labialis. So herpes simplex labialis associated urticaria was another observation, but it presented very strikingly. It wasn't chronic urticaria or difficult to treat, but it was catchy in the sense that recurrent unilateral facial urticaria. My next patient, young male patient, 20 age, chronic urticaria for 1 year and not related to any obvious reasons that I could detect clinically. And most of the investigation, as we all know, that turned out to be futile. And I was again back to square one, giving only a updosing of antihistamines and whatnot. Various second generation antihistamines were added. Intermittent doses of corticosteroids, some of the doctors have prescribed him. Partially he responded, but he never used to be completely happy. He required continuous administration of antihistaminic agents. So relapsing intermittent exacerbation was something which worried me, and he gave an important history at one of the episodes. Doctor, can it be related with, I get little abdominal discomfort And then suddenly I could say that now it's going to shower on the skin for multiple weeks. So acute diarrhea before the onset of disease was one history. Second history was acute intermittent exacerbation. And there was recurrent history of watery diarrhea and abdominal discomfort. With this background, I thought again of working this up for some sort of GI infection. He was worked up for microbiological investigation. The stool as well as the culture suggested the Yersinia infection, and which was, I was not very sure whether it is really causing urticaria. So as per the gastroenterologist suggestion, I prescribed him a course of ofloxacin for 7 days, and antihistamine I continued. And gradually, I realized that the need of antihistaminics was much low. Initially, 2 or 3 times of updosing used to be required. He was well controlled with a single dose after the course of ofloxacin, and gradually, surprisingly, even without antihistaminics, he did very well for the next 6 months. So Yersinia infection, I was wondering whether this is associated with chronic urticaria. In fact, the patient was very happy to receive a course of antibiotic along with antihistamine, and finally he was weaned off the antihistamine. So that was a good this thing. There was one or two small references, not big studies, but it is said that altered gastroduodenal permeability is associated with allergic or pseudo-allergic reactions in the patients who are susceptible for chronic urticaria. And my last one, a young patient again, who used to have recurrent acute urticaria. But he used to say, doctor, this is a summer allergy. And I used to say, what kind of summer? And you have suffered so many summers. You have faced so many summers in your life. Why only 3 years you are having this summer allergy? 3 successive years. As soon as you started getting, heating in the month of February to March, and up to May to June, it used to last for the urticaria. On clinical examination, there was pityriasis versicolor and malassezia folliculitis. These were the repeatedly associated things I could visibly see along with the urticaria, as you can see over here. So I gave him the initial antihistamine treatment, and I just wanted to observe. So when he came for the next year, he could actually literally point out that, see, doctor, these are the eruptions which are coming before the onset of itching. And careful listening to the patient helps sometimes. So there was a dramatic resolution of urticaria after the treatment of versicolor, along with the systemic antifungals and topical antifungal treatment. So malassezia infection associated with chronic urticaria is one thing that struck me. I must thank Marcus. I discussed this kind of patients with him, and at one of the ESPD meetings that if Marcus, do you remember? If you remember, then you had said that you compile all the clinical presentations and then you do this kind of workup, and it was really very helpful. And I must thank Marcus for helping me to for this observation to put it on the paper. Thank you, Marcus. Thank you so much. So my key message over here, in the patients with recurrent urticaria, acute exacerbations are something which are striking, and a detailed history and clinical examination is must for preceding or during the episodes. There could be actually a clinching evidence of underlying focus of the infections, and appropriate treatment may be curative. So you must try this. However, such experiences, of course, are rare, and but rarity that makes it unique. Thank you very much. Marcus, if you see, these are the memorable pictures with you. And thank you for teaching us and inspiring us for working in the field of urticaria. Marcus and Zuberbier, it's a wonderful time with you at Berlin. Thank you so much.
Thank you, Vijay. That was an excellent presentation. In fact, Vijay Zawar is a true astute clinician. He has so many cases, and we have published few. Vijay, you can stop sharing your screen because this is interesting. In fact, Well, obviously, tinea epidemic is going on, and that is one of the reasons where you see urticaria. This thing. I think Marcus, happy you have to be here. I thought this thing. Marcus, any of your suggestions or comments for Vijay's observations?
No, it's always nice to be in touch with you. And look, we learn from each other. We all see a lot of patients, and that brings with our conversations a lot of experience that should be shared and must be shared. So thank you very much. Thank you also for your kind words. Look, it is very different to talk to, let's say, GPs, specialists, or urticariologists such as yourselves, no, or ourselves, no. And we will see rare patients. We will see very interesting cases, and that is not to say that this doesn't happen in the with our GP friends or with specialists out there. But we must keep the open eye, and we must be thinking outside of the box every once in a while when the standard does not apply. And thanks for doing that, Vijay. Beautifully presented. Thank you very much.
Yeah,
Thank you, Marcus.
Very nice. In fact, we do see the cases. Abir, have you seen tinea and urticaria? Because you have been forefront for our ITASA. Your distinct points.
You're muted. Abir, are you muted? Okay. Yeah.
No. Can you hear me?
Yeah.
Yes.
Yeah. So we had Dr. Zawar himself, after talking to him a few years back, and then maybe patients started coming. And yes, I have also certainly noticed a few number of patients who keep getting urticaria, and you treat, they have typically got chronic or recurrent tinea, and you treat it and the urticaria goes away. I remember at least 2 patients offhand that I saw this, yes, certainly. And after talking to Dr. Zawar only, probably I started really looking for this, and I found it.
Thank you, Abhi, for your experience.
Yeah. Thank you. So obviously, all these observations, in fact, been published, and I request Vijay must publish these observations so that everybody is aware that scabies, tinea, herpes also can trigger urticaria, and which are very common in India. And now the fungal epidemic is on, so obviously that could be a cause. Thank you for these words. In fact, this has been a truly enriching experience for all of us. First, the presentation by Marcus, then the newer things, and then by Dr. Vijay's presentation and a panel discussion, which gave us too many messages, which would, in fact, I take him as a stay-home messages. You stay at home and keep these messages and improve your practice, and that is what we are to. You learn from others, we learn from our gurus, and give best to the patients, and that is what gives you satisfaction. So with this, I thank all of you, Dr. Marcus, Dr. Abir, Dr. Vijay, Dr. Krupa, and Minarini, for giving this a platform for sharing our knowledge and expanding our horizons and giving best to the patients. Well, we hope that this will be continued similarly. We have got 2 more these sessions in next month, which all of you are requested to join to learn more and more about urticaria. Thank you very much. Over to Mr. Sanjay.
So thank you, sir. Thank you very much, Dr. Kiran Godse. I mean, it was a very engaging session, and I can share with you more than around 370 doctors all India have joined on a day when we have at least half a dozen webinars going on.
Oh, really?
Among dermatologists. So, I mean, looking into the, the so much of deluge of, I will say, of webinars, you have still 370. It's a big, and doctors have joined from Silchar to Gorakhpur to Mumbai to Goa, everywhere. I mean, north to south and west. So, and I'm sure, looking at the faculty that we had, Dr. Marcus Moore, who is expert in his own domain, and we thank you, doctor, for being with us for this engaging Urticaria Decoded session. We also thank Dr. Kiran Godse, who has been, who is an Indian expert and also international expert on urticaria, and has always been helping us to conduct such meetings. And we have Dr. Abir Saraswat. Thank you very much, sir. And thank you, Dr. Vijay Zawar also, and Dr. Kripa Shankar also. I also take this opportunity to thank all the people who have joined from across the country, taking their time off on Sunday evening and to be with this. And we will request, as Dr. Vijay also said, there is a session, 2 more sessions. Dr. Kiran already said there are 2 more sessions planned in August, and which will be equally engaging. So please join us and be with us. So we have already introduced our international brand of Blackstone in India. I'm sure we will be shortly reaching out to you for your experience with our brand. Thank you very much. Thank you, sir. Thank you, all of you.
Thank you. Bye-bye.
Bye-bye.
Bye-bye, sir. Bye-bye. Thank you all.
Thank you, Marcus, Kiran. Thank you. Bye.
Bye-bye.
Abir, Krupa,
Bye-bye.
good day. Bye.